penguin, and its primary prey is the Antarctic krill. So I'm just going to go through these briefly. What I want to talk about with these models is the approach that was used to set them up and the model structure. And it's really the model structure, because as Beth said, when you really get down to coding these up and looking at them, a lot of what you do with the invertebrates and the vertebrates are actually the same structures, it's just what you call them when you set them up. And then talk about the limitations of these, and then I said the three examples are Calmus and Marchicus, and then the Adelie. All right, so the first example is from a paper by Charlie Miller that was published in Fisheries Oceanography a while ago. And what they were trying to do here is examine the Gulf of Maine, which is off the east coast of the United States, and looking at the distribution of Calmus and Marchicus in that area. And it turns out that's a very important prey item for cod and haddock, and an important part of their diet. And they're trying to figure out where the sources were, because the populations of Calmus and Marchicus over Georges Bank are not resident populations. They have to be brought in from someplace else. Right, so the thought was, well, they came up here off of Canada, coming out here in the Gulf of Maine. All right, so that's sort of the setup. And Beth just said you set these models up within a context of questions that you want to answer. And that was the answer that was the question that they were looking at here. So they had some various places in here where they thought the zooplankton might be coming from, and that's what they wanted to test. All right, now the individual-based model is structured around the life history of the animal. And as Beth mentioned before, you have to understand the animal or the organism that you're trying to model here. And here the animal's life history is providing the framework. All right, there's an egg, these various juvenile copepidid stages. This animal goes into diapause, which is a resting stage, part of its life history. And when it does that, it goes deep in the water and hangs out during the winter, comes back out as it turns into an adult, and then the adults spawn in the surface waters. All right, so this life history defined the structure of the model, and each of these stages has different attributes, different characteristics, different behaviors, biology, that kind of thing, and they use the available data to parameterize these different stages and the different attributes of the stages. And then when you do that, you end up having to track those attributes in space and time. All right, so they set up this, as I said, an individual-based model to look at this and go through these different stages. All right, now, as I said, there are attributes, and what's shown here are the vectors, if you will, or the attributes of each of the stages. I just picked a few of them. This is the diapause stage, the resting stage. And these are what are tracked for these animals, okay, whether it's alive or dead, whether it's inactive or ready to molt. Right? And that's easy to say, but when you try to write the equations or the algorithms to decide whether the animal is going to be still in diapause, what cues it to come out of diapause, and then if it molts. There's a lot of information, algorithms, or whatever behind each of these. And then these are just tracking where it is in latitude, longitude, and depth. And that's important because in this model, the cue coming out of diapause was assumed to be the change in the light as you go from winter into spring. All right, so that's the resting vector. The female part of the model, all right, you have to know the age since the female matured so that she's ready to produce eggs. All right, whether or not her clutch or her eggs are ready to be released, and then knowing where it is in space in X, Y, and Z coordinates. All right, and then again, the age since maturation seems easy to say, but you have to find a way to calculate. All right, this animal became an adult here. Now she's maturing, and now she's ready for spawn. Right, and then the nuclei here, right, going through what stage it's in, whether or not it's ready to molt between stages, and again, sorting all that out. But these are the variables that have to be tracked for every individual in this model, right? And so each of these is tracked and kept up with doing the calculations. So these kinds of models tend to be computationally intense because of this, and this is just, and this was not a particularly difficult one. And one of the criticisms I hear a lot of times with these individual-based models is, well, so what, you can't really verify it, you can't really do anything with it, but you really can't, because you have these kinds of data here, all right, where you verify the population structure and timing. Right? You might not be able to get absolute numbers, but you would certainly know that the eggs from observations, and here you use sort of a whole range of kinds of things. You know that the eggs should appear at a particular time because you see the different stages, and this is based upon observational data. And so you can, in fact, verify this kind of model through using this kind of information about when things should be appearing, the time period it takes to go from an inoculate into a copipidid or a juvenile into an adult. There are ways to look at the verification of these and know whether or not those algorithms you put for each of these components of the model are in fact producing the right kind of results. All right, so this is one way to do it. This is in time, the number of days from the solstice, which is what they're using in this particular model, is the cue for the animals to come out of diapause, and going over this period when you get to reproductive events. And the other way to do it is this is a spatial distribution. Because this is all time, they also have to have a circulation model to move these particles around. And this is the spatial distribution of the life history stages, all right, where the yellow ones are the adults, the blue ones are the eggs and octi, and the copipidins and things. And you can see this is on one day of day 160 and day 190, 30 days later. And you can verify, again, the model by the distribution of where these animal stages are in this particular area. All right, so this is again, computationally intense way to do it, or these models tend to be computationally intense, as I said, because this is embedded in that circulation model. All right, the limitations of this particular model, all right, is that the growth and the reproduction of this animal was assumed to be controlled only by temperature. And that works for a lot of things, but we also know that things like food quality and food quantity are important to the growth. And in this particular model, that wasn't included. And Beth mentioned things about knowing the diet, which I'll come to here in a minute. It assumed that light control emerges from diapause. We don't know really what controls things like that. And other animals have different behaviors, in terms of activity that are related to seasonal cycles. And in this particular case, there's not any feedback between the zooplankton and its environment. It's not extracting food from the environment or giving food back. All right, so what Beth had just showed is a way in which you interact the individual-based kinds of models, which you tend to have for the higher trophic levels with the environment. And this is a second paper in the same volume by Francois Carlani. Again, focused on columns, been marching this up here, but he attached it to an ecosystem model. And he retained all the life history structure up here, and the different stages interact with this ecosystem model in different ways, because, again, each stage has its own needs and its own attributes, and how it accesses this rest of the ecosystem is dependent upon what those are. All right, so if you take that and blow it up, now the model looks like this, where there is this ecosystem model over here, or lower trophic level model, more correctly, with the phytoplankton, microzooplankton, and detritus. There's all this detail over here for the individuals, for the particles. And in this case, the individual particles are actually combined to form a population because it's using what's called the Lagranglan ensemble particle, where each particle is a whole group of individuals with some variability associated with them. Right? An important thing in this one is that there are multiple food sources, multiple diet, or different diet structure for the different stages of this particular zooplankton, and also tracking the lipid. All right, so this is an example of, you don't just eat the food, you're needing a particular type of thing from the food, in this case are through ingestion, through egestion, and also through mortality, right? And that's going to be the same, even though this is a zooplankton, that's going to be the same for just about any animal. You know, it has to eat, it has waste products that go back into the environment to detritus and nutrients, and then the mortality ends up going back into the environment, into the lower trophic levels in this case. So these are the, if you have one of these models, these are where you have to have the connections that go into these larger models. And the Atlantis model that Beth was just showing has these same kinds of connections between each of those bigger, each of those members of the higher trophic level and the lower trophic level environment, which means these are where you need to put some of your information, or this is where you need to put your effort in being able to define the connections in this model. Okay, model setup, right, again, it's these vectors of looking at the attributes of the particles. All right, this is just one of them. It's based on, again, based on the life history stage. It's looking at the mortality of individuals in the particle. This paper has a huge table in it where it gives all the attributes of the particles. The reason I put this up here is not because I expect you to really look at it, but if you look at this, there are not any equations here. It's if this, then that, if this, then go there. All right, so once you get to this level of detail in these models, you don't have an algorithm. You're doing testing all the time that if it's bigger than this, then you go there. If it's smaller than this, then go here. All right, and that's an issue with these kinds of models because that kind of structure doesn't work well on parallel machines. All right, and so they're not algorithms in the sense that we have governing equations or some type of equation that's easily applied across all of this because it's all different when you go through your testing with different situations. Right, and that's particularly when you start adding in things like lipid, carbohydrate, and protein in terms of food quality. You want this much carbohydrate, but it has to be in the ratio that's appropriate for the lipid and the protein. All right, so there's a lot of testing that goes on to do that. All right, the results from this model, you can do things like this is the phytoplankton concentration, and because there is a link directly between the zooplankton, each of the stages, and the phytoplankton, you can in fact estimate grazing control on phytoplankton blooms. And you could do that in the aggregate kind of models that we heard about yesterday, but in this case you can say it's the adults doing it, not the smaller ones, or vice versa, and you can apportion what is actually controlling the distribution of this phytoplankton bloom. You can also look at the characteristics of individual particles, which is shown here, estimating the growth, where it is in time, its migration, looking at its reproduction, and the survival. Right? So this gives you 4 examples of the particles that were analyzed to look at those characteristics. And then you can take the Lagranglan ensemble particles and turn it back into a biomass. And here this is the distribution of the zooplankton biomass in this model. So when you start combining the Lagranglan and the Eulerian, which is what the words are. The Lagranglan is that you track something in space and time. Eulerian is you sit at a point and look at what's going on. So in this case, what this has done is combine those 2 approaches to modeling, and you get a larger variety or a better look at what's going on. Right, but the limitations are the food quantity and quality. This particular test shows that those are really important, but the focus here is only on lipid. There's also carbohydrate and protein that are really important for somatic growth. Right, and in this particular model, these were assumed to just be there in the quantity